The overall goal of this procedure is to culture thymic medullary epithelial cells, or mTECs, in a three-dimensional organotypic culture, or OTC, system. This is accomplished by first placing the fibrous scaffold material into a 12-well filter insert, followed by the layering of a freshly prepared fibrin gel containing human skin fibroblasts. After 4 to 5 days of pre-culture, the mTECs are seeded onto the scaffold and cultured under submerged culture conditions. Ultimately, immunohistochemistry, RNA isolation, and FACS can be performed on the co-cultures to assess the development of the seeded mTECs.
The main advantage of the 3D organotypic co-culture systems versus the existing 2D culture systems is that they are far superior in recapitulating mTEC differentiation, proliferation, and the maintenance of promiscuous gene expression. Today, demonstrating this technique will be Iris Martin, a technician from our collaboration laboratory.
When the human dermal fibroblasts reach confluency, prepare the scaffold fibers by boiling, drying, and ironing the 0.4 to 0.6 mm thick pieces of viscose nonwoven fibrous material. Use a metal hole punch to cut them into 11 mm circles. Place the cut fibrous material between 2 slides, wrap the slides in foil, and autoclave them. Once sterilized, place the inserts into wells of a sterile 12-well plate, and then each scaffold into a corresponding insert. Next, mix 2.7 × 10⁻⁴